Hey, it's Maddie, and let's talk about pharmacology. Before everything else, just a little bit of introduction. I am Adine, a 21-year-old registered pharmacist from the Philippines, and I am currently a first-year medical student. Let's start by defining what is pharmacology. When we hear a word with the suffix of -logy, we automatically think that this is a study of something. In this case, by definition, pharmacology is the branch of medical science that deals with the study of the fate and mechanism of drugs, its therapeutic effects, and the adverse reactions associated with it. So there are actually a lot of branches of pharmacology, but we are only going to discuss the three main branches. First one, we have pharmacodynamics, emphasis on the letter D, because pharmacodynamics talks about what the drug does to the body. The word drugs comes first. Next to that, we have pharmacokinetics, which refers to what the body does to the drug. If you're a Filipino like me, to remember this, you put the emphasis on the letter K, which would stand for the word katawan, the Tagalog translation of the word body. Body comes first. The last main branch is the pharmacotherapeutics, which is the study of rational drug use in the management of certain diseases. We have heard the word drug so much already. So what are drugs really? Drugs are any article or agent that is used for the mitigation, diagnosis, prevention, treatment, and cure of diseases in man and animals. Meaning, even the agents or articles that are given to our pets are also considered as drugs. Another definition is that drugs are articles other than food that elicits changes in the physiological and anatomical function of the body. Now, let's talk about the four classifications of drugs. Number 1, we have functional modifiers. From the word itself, modifiers. This classification of drugs acts by altering the body's normal response. For example, we have antipyretic drugs. They lower the body's temperature in the events of fever, wherein fever is the body's normal response during infection. Second classification are replenishers. This is used to supply what is insufficient. For example, are electrolytes and insulin. Third are the diagnostic agents, aids in diagnosing certain diseases. For example, we have edroponium, which helps diagnose myasthenia gravis, which is a neuromuscular disorder. Last classification are the chemotherapeutics. So when we hear the word chemo, we immediately think about anti-neoplastics or anti-cancer drugs. But antibacterials, antifungals, and antivirals are also under this classification because by definition, chemotherapeutics kills or inhibits growth of cells considered foreign in the body. Let us now focus on discussing the first of the three main branches of pharmacology, which is pharmacodynamics. As what I have said a while ago, pharmacodynamics is what the drug does to the body. By proper definition, it is the study of the biochemical and physiologic effects of drugs in biological systems and the mechanism by which these effects were produced. Under this, we are going to be talking about the mechanism of action of drugs, drug-receptor interaction, and dose-response relationship. Let's focus first with the mechanism of action of drugs. This is divided into two topics, namely target protein-mediated and non-target protein-mediated. Let's start with target protein-mediated. Under this, we have subtopics. First, structural proteins. Under this, we have the microtubules and tubulin as examples, which are organelles within the cell that helps in maintaining the framework of the cell. As per drugs, these said proteins are inhibited by the taxanes, bincalcaloids, colchicine, and griseopulpine as examples, wherein this drug's mechanism of action has something to do with disrupting the assembly or function of microtubules and tubulin. Next subtopic we have are the regulatory proteins. This is divided into four classifications. Voltage-gated channel. By word voltage, this has something to do with conducting changes in electrical signals. For example, are sodium channel blockers, potassium channel blockers, and calcium channel blockers. As you can see, these are all related with electrolytes. Second, we have the carrier molecules. In this classification, we have the sodium-potassium ATPase pump and potassium proton pump. Next, we have enzymes, which are biological catalysts that acts by shortening the biological reaction. And here we have phospholipase A2, cyclooxygenase, lipooxygenase, and monoamine oxidase as examples. Last classification we have are the receptors, which are macromolecule cell components that has a specific stereochemical configuration where a ligand binds, usually in a lock and key mechanism. Let's discuss further the four types of receptor. Let's start with type 1, ionotropic receptor or ligand-gated ion channels. Located in the cell membrane, onset only takes milliseconds. Examples are nicotinic receptors, GABA-A receptor, glycine receptors, and 5-HT3 receptors. Type 2, also known as metabotropic or G-protein coupled receptors, also located in the cell membranes, onset takes seconds. This is divided into two kinds. Number 1, secondary messengers, wherein we have cyclic adenosine monophosphate, cyclic guanosine monophosphate, inositol triphosphate, and diacylglycerol as examples. Number 2 are G proteins, which is further divided into three. G sub S, which stimulates adenylyl cyclase, the enzyme that synthesizes cyclic adenosine monophosphate from ATP. Example are the beta receptors. Next, G sub I, which inhibits adenylyl cyclase, which then results to decrease in cyclic adenosine monophosphate. Examples are the presynaptic alpha-2 receptor and muscarinic 2 receptors. Last, G sub Q, which stimulates phospholipase C that converts the phosphatidyl-inositol 4,5-biphosphate into the second messenger inositol triphosphate and diacylglycerol. Examples are alpha-1 receptor, muscarinic 1 and muscarinic 2 receptors, and postsynaptic alpha-2 receptors. We are now done with type 2. Let's head on to type 3, also known as kinase-linked receptors. Still located in the cell membrane, onset takes minutes. Examples are insulin and growth factor receptors. Down to our last type of receptor, type 4, also known as gene transcription linked receptors or nuclear receptors. This one is different because it is located in the nucleus. Onset takes hours because this bypasses the cell membrane. Examples are steroid hormone receptor, thyroid hormone receptor, vitamin D and vitamin B derivative receptors. Lastly, under the mechanism of action, we have the topic non-target protein mediated. This means that this does not have any binding site or receptor but still elicits effect. Under this, we have colligative mechanism, which has something to do with solute in the solution, because colligative means bound together. For example is mannitol, which is used as osmotic diuretic and also to decrease a patient's intraocular pressure in the events of cerebral edema. Next, direct chemical interaction. Under this, we have the neutralization reaction, wherein acid and base react to form water and salt. Also, under direct chemical interaction, we have chelation, a method for removing heavy metals, wherein a chelating agent binds with a heavy metal, which would then produce a product that would be readily excreted. Lastly, we have the counterfeit mechanism, wherein we incorporate an imitation of a certain biological compound that produces opposing effect. For example are antimetabolites, which are purine and pyrimidine analogs. Now we're done with the mechanism of action. Let's head on to the drug-receptor interaction. Let's start with the features of the ligand. Number 1, affinity. It is the ability of a ligand to bind to a receptor. Think of it as a metal and magnet interaction. The metal does not change its conformity, nor does the magnet. It just binds together. Meanwhile, when we say intrinsic activity, this refers to the ability to elicit response. Second consideration is the classification of ligands. We have two classifications, agonist and antagonist. Theories refers to the possibilities of ways on how the ligand binds with the receptor and the way it stimulates response. Let's start with agonist. By definition, it is affinity plus intrinsic activity. It is further classified into full, partial, and inverse agonists. With full agonist, intrinsic activity is equal to 1, 1 here is our whole number, meaning it produces all of the expected effects from a particular receptor. With partial agonists, intrinsic activity is greater than 0 but less than 1, meaning it produces some of the expected effects, and the effects it produces is mixed agonists and antagonists. Lastly, with inverse agonists, intrinsic activity is less than 0, meaning this so much acts as an antagonist rather than an agonist. Let's proceed to antagonists, which is further classified based on mechanism of action, duration of interaction, and surmountability of interaction. Based on mechanism of action, antagonists are divided into 3 groups. 1. Functional antagonism, also known as physiologic antagonism, refers to 2 ligands acting on different targets, producing opposing effects. For example are acetylcholine and norepinephrine, which acts on 2 different receptors, namely muscarinic 2 and beta-1 receptors respectively. Acetylcholine acts as agonist when bound to muscarinic 2 and produces the effect bradycardia. Meanwhile, norepinephrine also acts as agonist to beta-1 receptor but produces the opposing effect of tachycardia. As they produce 2 opposing effects, they antagonize each other. Second group is the receptor antagonism, also known as pharmacologic antagonism. It is when 2 ligands act in the same receptor but produces opposing effect. For example are norepinephrine and metoprolol, which is a beta-1 selective blocker. Both act in the same receptor, which is beta-1, but produces 2 opposite effects. Norepinephrine acts as agonist that would then result to tachycardia. Meanwhile, metoprolol blocks the beta-1 receptor that results to bradycardia. Third group is the chemical antagonism, refers to the process of neutralization and chelation. Let's proceed to based on duration of action. This is only divided into 2 groups, reversible, which lasts about less than 24 hours and is formed by non-covalent bonds such as van der Waals and hydrogen bonds, and irreversible, greater than 24 hours and is formed by covalent bonds. The last classification is based on surmountability, which is also divided into 2 groups. Competitive, which means that it overcomes the effect of antagonists by increasing the dose of agonists. This interaction is surmountable. Non-competitive, which means that it does not completely surpass the effect of antagonists by increasing the dose of agonists, which makes this interaction non-surmountable. Let's now head into theories. Number 1. Hypothesis of Clark states that maximum effect is achieved when all receptors are occupied. Number 2. Hypothesis of Peyton, or rate theory, states that the maximum pharmacologic effect does not depend on the occupancy of receptors but on the stimulus. Last is the hypothesis of Arians and Stephenson, or occupancy theory. This states that effect lasts as long as receptors are occupied, which means that the number of receptors occupied does not matter. As long as there are receptors that are occupied, the effect is elicited. We are now down to our last topic for pharmacodynamics, the dose-response relationship. This topic is divided into graded dose-response relationship and quantal dose relationship, as seen here in the outline. Let's start with the graded dose-response relationship. The plot of the graph focuses on the degree of response versus the dose or the log dose. As you can see in this graph, the y-axis stands for the degree of response and the s-axis refers to the drug concentration. We have 4 parameters present in this curve. One is efficacy, or the maximum achievable response. Second is the ceiling dose, or the smallest dose that produces the maximum response. Third is the potency, or the dose needed to achieve the 50% of maximum response. And lastly, the slope, or the degree of changes in response with a change in dose. This graph is applied in measuring the different characteristics of a drug. First is in measuring the efficacy and potency. Efficacy refers to the maximum achievable dose. Meanwhile, potency is the measure of drug activity expressed in terms concentration of drug that can elicit effect. If we are to interpret this graph, drug X and Z has equal efficacy because the curve is of the same height, meaning they have the same maximum achievable dose. And as per drug Y, it is the least effective because its curve only reaches half of the curve of the other drugs. And when we talk about potency, drug X being most potent because it elicits effect earlier than the two other drugs, which then this would mean that drug Z is the least potent. Another application would be for comparing the full, partial, and inverse agonists. And the last application is for comparing competitive and non-competitive antagonists. As you can see, the curve for competitive antagonistic drug is shifted to the right and is of the same height as the curve of the drug alone, because as I have mentioned a while ago, for competitive antagonistic drugs, it can overcome the effect of the drug alone just by increasing the dose. And the curve for non-competitive antagonists is shifted to the left and has a lower height because this does not completely overcome the effect of the drug even by increasing the dose. This is the last subtopic under the dose-response relationship, the quantile dose response. The y-axis here stands for the cumulative number of patients responding, and the x-axis still refers to the log dose. We have four parameters present, namely ED50 or median effective dose, which refers to the effective dose in 50% of the given population, TD50 or LD50, or median toxic dose or median lethal dose, which refers to the toxic dose in 50% of the given population, therapeutic index, which refers to the measure of relative safety, which is computed by dividing the median toxic dose with the median effective dose. Margin of safety refers to the doses between the effective dose and toxic dose that is still safe for the patients. And that is it for pharmacodynamics. This discussion focused more on concepts, as what you have watched, I did not mention a lot of drugs yet because it is important that you understand first these said concepts before applying it to the specific groups of drugs. Again, this is Maddie and we have talked about pharmacology. If you guys have any questions or clarifications, you can email me on the email address stated in the description box and I'll try my best to answer. Thank you guys. See you on my next video for the discussion of pharmacokinetics. Please like and subscribe!